And now next, we're going to hear from two of our MITO2i graduate scholars. And we'll start with Jacob Smith from Melika's lab, which will be presenting on assessing mitochondrial dysfunction due to SARS-CoV-2 infection in a heart-on-a-chip platform. Followed by him will be Fatima Mishafe, PhD candidate in Dr. Jason Main's lab at the Hospital for Sick Kids and in the Department of Biochemistry at the University of Toronto. She will be talking about mitochondrial metabolic therapies for heart disease based on regenerative potentials of pediatric heart. Jacob, over to you.
Hi, everyone. I'm Jacob Smith. I'm a PhD student in Professor Radicic's laboratory, and I'll be talking to you today about assessing mitochondrial dysfunction due to SARS-CoV-2 infection in a heart-on-a-chip platform. So just some introduction about the heart-on-a-chip platform we're using. It's called the BioWire2, and it is a 3D cardiac tissue organ-on-a-chip platform. So it's composed of cardiomyocytes and fibroblasts which are combined together in a hydrogel matrix. This matrix is then seeded into a well of a small polystyrene chip with two wires on either ends. The tissue, after a few days, it begins to compact and it attaches to these two wires and then forms this sort of dog bone-like structure, which you can see on the right-hand side there. The tissue then begins to spontaneously contract and starts to kind of beat and pull on these two wires. So using a MATLAB kind of program and some manual measurements, we can extract the tension from this tissue and figure out the amount of force that is being generated every time that it contracts. We can also use calcium-sensitive dyes to measure calcium flux within the cardiomyocytes. On the bottom left, you can see some of the measurements we use to calculate the force of the tissue, as well as a kind of zoomed in schematic of the actual fluorescent wire that are used to, that are deflected as the tissue contracts. To the right of that, you can see our kind of force output graph. So the peaks in this graph represent contraction events of the tissue kind of pulling together. And then to the right, you can see kind of a more broad schematic overview of the actual platform itself. So the polystyrene chip with the well and the tissue in it, and then the kind of assessment chamber we would put in a microscope. So moving on to some preliminary data that we've obtained for this project so far. So early on, my group found out that upon infection with the SARS-CoV-2 virus, we found that our BioWire tissues had attenuated tension during contraction events. So you can see on the graph there just to the right, in a control tissue, we see kind of nice peaks of tension as the tissue pulls together, but upon infection with the virus at MOI5, we see like almost complete attenuation of any sort of contractile peaks at all. So it's very interesting to see how much the virus was able to lower the tension in our tissues. We've also had the opportunity to do some TEM microscopy of infected cardiomyocytes, and we found that these cardiomyocytes have disarrayed sarcomeres and enlarged mitochondria. So near the bottom of the slide, you can see an image there kind of a control sample of some cardiomyocytes. And you can see that the sarcomeres look very smooth and aligned, and the mitochondria are relatively normal looking. And then directly to the right of that, as you can see, it's labeled, there's quite a bit of sarcomeric disarray so that they don't look kind of normal and they're all no longer aligned, as well as the mitochondria are significantly larger than the control sample. With further magnification, we're able to see that the cristae are disoriented in the mitochondria, perhaps indicating that they're not functioning as normal due to their strange shape. We have also done some preliminary RNA sequencing, which has revealed that oxidative phosphorylation is the most inhibited pathway in our system, perhaps indicating that the mitochondria have become dysregulated and the energy systems inside our cardiomyocytes have switched to other methods. So moving on to the specific aims of my project, there's three of them. And the first is to develop an improved analysis system for our platform. So as I mentioned earlier, to calculate these tension events during contraction, we're using a MATLAB-based system, which requires a fair amount of manual analysis and measurement. So what we'd like to do is use computer science techniques and machine learning and some computer vision to develop a new analysis platform that can just read videos of our contraction events and then quickly output data showing these tension changes. And this would be helpful for us because it could greatly increase the amount of throughput of our system and let us test a lot more samples more frequently. The second aim is starts with infecting the actual biowire tissues with the SARS-CoV-2 virus using the University of Toronto CL3 facility. So we would infect the tissues, and then for every day for 2 weeks, we would be monitoring the tension and the health of the tissues to see how it's changed. And then at the end, we would be performing genomic and proteomic analysis to investigate differences in oxidative phosphorylation within our cardiomyocytes in our organ-on-a-chip platform. The third and final aim of the study would be to actually look at how SARS-CoV-2 impacts the actual mitochondrial dysregulation. And we would do this by measuring the oxygen consumption rate and the extracellular acidification rate to understand changes in metabolism. We would also like to look at calcium handling and ROS production to see how that changes within our tissues. One important aspect of this project is how interdisciplinary it is. So it combines several different disciplines together, which are required to kind of run this project. Specifically, it uses cardiac tissue engineering, computer science, virology, and mitochondrial biology kind of together as one unit. Tissue engineering is really important to the actual biowire design and manufacturing. Because this is a complex organ-on-a-chip platform, so knowledge of cardiac tissue engineering is required to use it. Mitochondrial biology and virology are also very important to understand kind of why the dysregulation is occurring, as well as knowledge about how to properly apply the actual SARS-CoV-2 virus to our tissues. And finally, computer science will be leveraged to actually design our analysis platform as a way to increase the throughput of our actual system using machine learning. And lastly, I'll just talk a little bit about patient engagement. It's important to engage with patients and kind of make the public aware of how dangerous SARS-CoV-2 is, as well as it would be interesting kind of to show how it can directly affect like the mitochondrial within people. And one of the ways we would performance outreach is by collecting serum from patients who have been infected with SARS-CoV-2. And then we would be applying this serum to our biowire tissues to determine if it has any changes in the health of our tissues or if it directly kind of influences mitochondrial dysfunction. And this is a really unique opportunity because my supervisor has access and authorization to infected patient serum at the UHN's Toronto General Hospital and St. Michael's Hospital. And lastly, I would just like to thank my lab and all the collaborators who have helped me with this project. And thank you for allowing me to present at the MITO2i Symposium.